Size exclusion chromatography is a technique used within biochemistry labs to isolate various proteins, amino acids, or fragments depending on their size. So essentially, for this technique, we are utilizing a polymerized agarose or acrylamide gel. And we can create different percentages, percent concentrations of the agarose or acrylamide, and depending on that percent concentration, we will have different pore sizes within our gels, okay? So essentially what happens is that we have this gel inside this column. This gel has pores. Now, normally, we have small pores within this gel, and we'll understand why in a second they're small. So, but right now let's talk about how these smaller molecules, they are able to enter the pores. Because the pores are small, we have these small molecules, they're able to enter. These larger molecules, on the other hand, are not able to enter the pores. Now, this may seem counterintuitive, but what happens is that since these smaller molecules are able to enter these pores, they are actually going to take longer to reach the bottom because they're in a way, they're taking the long way to the end. They have to go all the way through, and then finally they'll reach the bottom. Whereas these larger molecules, as soon as they enter, they're going to go really fast down because they cannot fit through these small pores. Okay, so they just pass straight on down. They don't have to take the long way down. Now, if these pores were big, then all of the molecules would be able to enter. These big ones, the medium ones, small ones, they'd all be able to enter, and that would not be effective. That is why we have small pores, because small pores limits only the small molecules from entering. Now, once again, size exclusion chromatography, it's based on size, and we utilize a porous polymerized agarose or acrylamide gel that has small pores. The smaller molecules will be able to enter the pores, and they will take the longer way down and exit last. The medium-sized ones, they'll be able to enter some pores, but they may not be able to enter all of them. So still they'll be a little lagged, whereas these larger ones, they will exit first since they do not fit into the pores at all.